Hello everyone, welcome to part 1 of 2 on our lecture on fats. So today we're looking at the storage, release, and the release of energy from lipids. So first, we're just going to discuss what fat cells are. So obviously, I'm assuming most of you guys have heard about fat or adipocytes as they're referred to. So adipocytes are large vacuoles that empty and fill depending on dietary intake and energy output. So these fat cells are essentially, we have them all in our body, and they basically shrink or increase depending on dietary intake and energy output. So we don't actually even when we burn calories or lose weight, we don't actually get rid of the cell. It stays there, it just shrinks. So from an article in Nature, the number of fat cells stays constant in adulthood in lean and obese individuals, even after marked weight loss, indicating that the number of adipocytes is set during childhood and adolescence. So this is where an issue where childhood obesity and being overweight as a child can become an issue, because this can increase that actual number of adipose tissues or adipocytes in your body, and I guess it can predispose you to essentially, even if you lost it, more weight gain later. And it's also why it's difficult for younger children who are obese when they become adults to lose the weight. But it is possible. So the storage of fats as triglycerides. So you'll see triglycerides commonly referred to as TGs. They're also sometimes referred as triacylglycerols, which then are also referred as TAGs. So adult humans ingest about 60 to 150 grams of lipids per day, which is more than 90% of triglycerides, which more than 90% are triglycerides. During digestion, triglycerides are split into glycerol and fatty acids, but following absorption, the products of lipid digestion are resynthesized into triglycerides and stored in adipocytes. So we need to break them down for them to move into our body, essentially. Then they're repackaged and stored. And here's a picture of a triglyceride, or the formula, sorry. So triglyceride mobilization. So there's two enzymes involved. This, and one example is the hormone-induced fatty acid mobilization in adipocytes. So one example here, we have the hormone epinephrine, which can stimulate a G protein, and we can use the enzyme adenylate cyclase. We can see we go through the pathways, we get a protein kinase A. This stimulates something called hormone-sensitive lipase, which is also known as HSL, and also MAG lipase. So the MAG lipase, again, essentially the lipase means it's essentially breaking down that fat into that fatty acid, and like we said, the fatty acid and a glycerol. So essentially this goes from a triacylglycerol to a diacylglycerol to a monoacylglycerol into the glycerol and fatty acid. So again, as I said, triacylglycerols are also known as triglycerides. So they're kind of synonymous. So essentially, hormone-sensitive lipase breaks down the triglycerol to diglycerol, as well as diglycerol to monoglycerol, and the monoglycerol is broken down into its individual constituents, fatty acid and glycerol. And again, this is rebuilt and then restored. So what happens to free fatty acids? So these are commonly referred to as FFAs after triglyceride mobilization. So free fatty acids move through the blood cell membrane of the adipocyte into the blood because it is lipid soluble. Then they bind to albumin, which is a protein in the plasma, and remember, it's not water soluble. It's lipid soluble. So once it's in the blood, it needs to be carried. Albumin carries fatty acids to tissues where they diffuse into cells and are oxidized for energy. So again, they can diffuse into the cell because they're lipid soluble. And as we talked about, our cell membranes have that phospholipid bilayer. They're able to pass through. So note, the brain, red blood cells or erythrocytes, so red blood cells, and the adrenal medulla, the top of our adrenal gland, or kidney, cannot use plasma-free fatty acids as for fuel. So glycerol. So glycerol is used to make more triglycerides. So again, we had the triacylglycerol, So as we talked about with a lot of stuff for monocarbohydrates and stuff like that, this indicates the number. So fate 1 of glycerol is that it's phosphorylated by glycerol kinase. So remember, kinase is involved in phosphorylation to glycerol phosphate. Then it can also be used to make more triglycerides. So here's the formula with the glycerol kinase. So we have our glycerol becoming glycerol 3P. So again, 3 is the carbon in the phosphate group. We can also have it formed in triglycerides, going from our glycerol 3 phosphate. So here's our G3P, using acyltransferase. So here we see another 2 coenzyme A's, like we saw in the citric acid cycle. Then this has phosphoiodate. Then we have a phosphatase reaction using H2O. Then we get our diacylglycerol, or DAG. And then we talked about acylcarnitine, and then going this way. So remember, the other way was the lipase, the hormone-sensitive lipase. But going this way is acyltransferase with the CoA, forming triacylglycerol. So again, glycerol can, and interestingly, glycerol can actually enter glycolysis or gluconeogenesis. So fate 2, or another fate, is that it can be phosphorylated by glycerol kinase to glycerol phosphate. It can be converted to dihydroxyacetone. So remember, that's in this step here where it splits. So there's G3P here and DHAP, or this is also referred to as DHAP commonly, dihydroxyacetone, acetone phosphate, sorry. And then this can enter glycolysis to generate ATP. So again, this can go straight in, and then we can go down and we can pump pyruvate, and then go through all the other cycles we talked about. So again, we can go glycerol. We need an energy to go from glycerol to G3P. And here we have another reaction with NAD plus to NADH, and then we get a dihydroxyacetone P phosphate. Whoops. So using fat for energy. It's simple, sort of. So essentially, we convert fat into that acetyl-CoA, which we know acetyl-CoA can be utilized to enter the citric acid cycle and then used in the electron transport chain. And this occurs in many tissues, especially the liver. So steps involved in using fatty acids as a fuel source. So first, we need fatty acid activation, where we create that fatty acyl-CoA. Then we need the transport of this fatty acyl-CoA into the mitochondria by carnitine. So we'll talk about carnitine quite a bit soon. And this is essentially used to get it into the mitochondria. That's my mitochondria. And then we have the conversion of fatty acid CoA into acetyl-CoA. And then, as we know, acetyl-CoA enters that citric acid cycle. So step 1, the activation of fatty acids. So fatty acids are hydrocarbon chains with a carboxylic acid tail. So here's that carboxylic acid tail in a hydrocarbon chain. So in order to become activated, the carboxylic acid is converted to a fatty acyl-CoA derivative in a reaction with coenzyme A. So remember, coenzyme A is the CoA. So this becomes that fatty acyl-CoA. So fatty acyl-CoA is a temporary compound formed when coenzyme A attaches to the end of a long chain of a fatty acid. This CoA is a 21 carbon molecule and is a long chain fatty acid equals 10 to 22. Therefore, fatty acetyl-CoA is 31 to 43 carbons. And it is a long chain fatty acid. So again, here's our fatty acid. Now we have fatty acid with our coenzyme A, and now we have fatty acyl-CoA. So our formation. So first, phosphorylation. We have phosphorylation of the fatty acid by thiokinase. So remember, kinase, aka fatty acyl-CoA synthetase. Then we have this forms the acyl adenylate intermediate. Here's our intermediate. Then we have a reaction with coenzyme A to form fatty acyl-CoA. So here we have going in. AMP is being released. We have the formation of our acyl-CoA. So some points about fatty acid activation. So here we can see our formula. The ATP, coenzyme A, and AMP, and 2 inorganic phosphates being produced. So that's why we get AMP, because 2 are being released. So here you see this gives free energy of 0.9, but the inorganic phosphate formed is hydrolyzed to 2P, So the overall gives free energy is negative 32.5. Fatty acyl-CoA synthetase is a different enzymes for short. So there's different versions of this enzyme for short, medium, and long chain fatty acids. So step 2 is the transport of fatty acyl-CoA into the mitochondrial matrix. And as we mentioned, this is heavily involved with carnitine. So the activation of fatty acids occurs in the outer mitochondrial matrix. But we still have to convert fatty acyl-CoA to acetyl-CoA, which can enter the citric acid cycle. The citric acid cycle occurs in the mitochondrial matrix, so the activated fatty acids have to get into the matrix. Carnitine is used to carry the activated fatty acids into the mitochondrial matrix, and fatty acyl-CoA is reformed once inside the matrix and then converted. So carnitine. Carnitine is synthesized from the amino acids lysine and methionine. So carnitine is also sold as a nutritional supplement in an attempt to increase fat oxidation. I'm not sure if this has been proven or not, but I think that you can tell that the goal, if it moves more fats into the mitochondria to be used, the theory is that by moving more fats in, it increase the use of fats and increase lipolysis. So activated fatty acids, or fatty acyl-CoA, will bind to carnitine to form fatty acylcarnitine. The fatty acyl group, or the fatty acyl-CoA, is transferred to carnitine, leaving the CoA behind. So here we have a carnitine molecule. And again, we can see that the CoA has been removed. So carnitine and fatty acyl-CoA equals fatty acylcarnitine. So here I have a carnitine molecule and our acetyl-CoA using the enzyme carnitine acetyltransferase. We can see that we form our fatty acylcarnitine with the removal of CoA and the bonding of these here. So the transport of fatty acylcarnitine into the mitochondria is a 3-step process. So 1, carnitine acetyltransferase 1, also known as CPT1, so carnitine acetyltransferase 1 enzyme associated with the cytosolic surface of the outer mitochondrial membrane catalyzes the formation of fatty acylcarnitine from fatty acyl-CoA. Second, we translocase an antiporter. We use the enzyme translocase, which is an antiporter in the inner mitochondrial membrane, which mediates the transmembrane exchange of fatty acylcarnitine for carnitine. So again, this is an important enzyme. So again, it exchanges the fatty acylcarnitine for normal carnitine. 3, within the mitochondrial matrix, carnitine acetyltransferase 2, so remember 2, catalyzes transfer of the fatty acid from carnitine to coenzyme A. So here's our antiporter mechanism right here, translocase. So first we have our first reaction where we have carnitine acetyltransferase, which is bound to the outer mitochondrial membrane. Coenzyme A is removed. Now we have our carnitine. Again, it's an antiporter, so if this carnitine is moved in and this carnitine is moved out, our acyl color, so this is our acyl carnitine, is moved in. Then again, another coenzyme A is used. We have our output using carnitine acetyltransferase 2, which is located in this inner matrix side of the mitochondrial membrane. And it's turned back into normal carnitine, which is then moved out and then used in the antiporter mechanism. So we can see here the outer mitochondrial membrane is permeable to activated fatty acids, but the inner mitochondrial membrane is not. Here's our inner mitochondrial membrane. So fatty acid activation to fatty acyl-CoA occurs in the cytosol. Then we have the transport across the outer mitochondrial membrane. Then we have the formation of fatty acylcarnitine. Here's our fatty acylcarnitine. Then it has transport across the inner mitochondrial membrane using the carnitine acylcarnitine translocase enzyme, like we talked about, through the antiporter mechanism. So here we can see going in here, and then this carnitine is being brought out and then being reused up here to form fatty acylcarnitine. Then we have resynthesis of fatty acyl-CoA in the matrix. So again, this fatty acylcarnitine is broken down, brought in, and then it's reformed here. And then the carnitine is taken out. So I guess it seems futile because you're breaking it down and then putting it back together to have the exact same molecule, but there's actually a purpose for this. So just to get it straight, first we have activate the fatty acid by converting it into fatty acyl-CoA through reaction with acetyl-CoA. Then we convert the activated fatty acyl-CoA to fatty acylcarnitine. Then we reconvert it to fatty acylcarnitine back to fatty acetyl-CoA. Yes, this occurs, but it is not futile, since the initial activation of the fatty acid is the trigger for the formation of fatty acylcarnitine, which brings about the oxidation of fatty acid for energy. So again, step 3 is the conversion of fatty acyl-CoA to acetyl-CoA, aka beta oxidation. So you'll hear this term quite often. So beta oxidation is FAD-dependent dehydrogenation. We can see here. Then we have a hydration reaction. Then we have NAD+ dependent dehydrogenation. Then we have thiolysis, which is a split of 2 carbons. Thiolysis through ketoacyl-CoA thiolase. And again, this continues until there's 2 acetyl-CoAs. So summary of fatty oxidation. So 1, there's the activation of fatty acids to fatty acyl-CoA. And this is a reaction between fatty acids and coenzyme A. Second, there's a transport of activated fatty acids into the mitochondrial matrix. This occurs via the carnitine, which involves the formation of fatty acylcarnitine and then resynthesis of fatty acyl-CoA once it's in the inner mitochondrial membrane. And 3, we have the conversion of fatty acyl-CoA into acetyl-CoA, which can then be used in the different cycles. Then this occurs in a series of 4 reactions, which occur right here. So this is beta oxidation is going from fatty acyl-CoA to acetyl-CoA. You can see here. Then, so again, it was 4 reactions that shorten the fatty acid chain by 2 carbons at a time. Then the final step produces 2 molecules of acetyl-CoA, which then enter the citric acid cycle. So again, the summary of 1 round of beta oxidation. So we have a fatty acyl-CoA with a FAD. As we talked about here, we need a FAD, H2O, NADH, and coenzyme A. So H2O, NADH, and our NAD and coenzyme A. Then our products are a fatty acyl-CoA, which is 2 carbons shorter, plus a FADH, because remember these are being, we can see here, dehydrogenations for FADH2 and NADH, and a hydrogen ion and acetyl-CoA. So, energy yield from fatty acid oxidation. So, for example, here's one fatty acid called palmitic acid, which is 16. We'll talk about the numbering soon, but this is palmitate, also known as palmitate. So, again, it costs 2 ATP to make fatty acyl-CoA. Although only 1 ATP participates, another is required for ATP resynthesis. So, a 16-carbon fat, like palmitic acid, can yield 8 acetyl-CoAs and 7 rounds of beta oxidation. So 7 rounds of beta oxidation would equal 7 FADH, 7 NADH, and 8 acetyl-CoA. So remember, we're getting this per 1. So this is 1 carbon, but if we had 16, we can see how we can get, but then with 16, we have to divide here to get these numbers. So again, we get 7 rounds. So anyways, keep going. So we get a 7 FDH, which remember is 1.5 ATP each, which equals 10.5 ATP. NADH, which yields 2.5 ATP each for 17.5. And 8 acetyl-CoA, which enter the citric acid cycle. And in 1 round of the citric acid cycle, we get 3 NADH, 1 FDH, 1 GTP. So that's 7.5 plus 1.5 plus 1 plus 10 equals 10. So 7.5 from our NADH, because remember it's 2.5. Then 1.5 for FADH, and then 1 for GTP equals 10. So then we got to add all these up. We get quite a good number. So again, so 7 rounds of, just from this one palmitic acid, we see we can get here 10, 17, and 10. So it's quite a bit of ATP. So this is essentially why fats are stored in sense in evolutionary wise, because there's such high yield when it comes to producing energy, it is some cost with a 2-ATP, but that's why they're almost stored as reserves, because they know if you survive on your fat, it produces a lot of energy. But yeah, that's kind of an evolutionary thing, but we'll continue our discussion on fats in the next lecture, so hope you guys enjoyed it and learned something. See you next time.